All right, so now let's talk about some of the specific cells in the immune system. We'll start off with natural killer cells. These cells, just like their name says, are natural-born killers. So they're almost like the silent spies or the James Bonds of the immune system. They have licenses to kill basically any cell, but hopefully they pick the right cells that are infected. And that brings up a question. What types of cells do you think they will go after? Well, I said one of them, virally infected cells and tumor cells. And these are really analogous to the CD8 cells, but CD8 T cells are more specific. The natural killer cells are non-specific killers. Okay? So the activity of these lymphocytes is enhanced by the cytokines interleukin 12, interferon beta, and interferon alpha. Just a tidbit there. So here we have a virally infected cell. We've seen this before. The virus gets chewed up inside the proteasome, travels to the RER, and makes its way to the top of the MHC1 receptor. Now, what was that subunit again that is the chaperone of a viral fragment? The beta 2 fragment, okay? See if you're paying attention. All right, so here's a natural killer cell and says, hey, this little piece of something that you have from inside you, cell, I think that is a problem. I think that might be harmful. So guess what? I'm going to perforate a hole in you, dump some granzymes in, and yeah, trigger off cell suicide for you. Okay? Bye. And so the granzymes trigger cytochrome C, which we already know triggers the caspase enzymes, and then they go and chew up some cell membrane stuff and other things, basically everything inside the cell, and boom, apoptosis. All right, so moving on now. All right, let's move on to some other types of cells, B and T cells. So B cells, when you hear B cells, what should you think of? Antibodies, right? They turn into plasma cells and make various types of antibodies. And those antibodies can go out and perform various functions. One thing is activating the complement cascade, which is a system of proteins that complements, it's probably how it got its name, complements the immune system to destroy all the attacking and offending agents, all the pathogens. Okay, other types of antibodies can trigger the allergic response. What types of antibodies do you think do that? IgE, and we'll talk about that whole process a little bit later. Okay, T cells. What do they do again? Well, you have to think about the two main types of T cells. The CD4, talk about first, we already mentioned it. They're the cheerleaders. They're the ones promoting propaganda to win the war against all those pathogens. So T cells, CD4 T cells, release cytokines and stimulate a variety of cells, macrophages, other T cells, and pretty much every type of immune cell. Okay, then there's another type of T cell, the CD8. Remember, it ate something. So those are the ones that are specific killers of virally infected cells and tumor cells. We will now discuss the differentiation, the activation, and finally the function of T cells. First, we'll start with the differentiation. T cells are born in the bone marrow, like other blood cells. They then migrate to a certain organ. Do you know the name of that organ? I'll give you a hint. It starts with a T. That's right, the thymus. Once in the thymus, these progenitor T cells undergo positive and negative selection. For positive selection, the thymus releases foreign antigens that mimic bacteria, viruses, or other pathogens, and watches to see if these baby T cells will react like they're supposed to. If they do not react, then they are apoptosed. If they do, they move on to the next step, which is negative selection. Negative selection is when the thymus releases antigens that mimic tissues in the body. If these progenitor T cells react to these antigens, that's bad. You don't want your T cells attacking your body, so they are apoptosed. If they don't react, then they move on to either CD4 or CD8 matured T cells. CD8 cells, as we've discussed, induce apoptosis of virally or tumor-infected cells. How do they do this again? What's this receptor that they interact with? The MHC1 receptor. And again, when a fragment of a virus or a cell that has undergone malignant transformation is on the surface, is being presented by this MHC receptor, that cell is bad and it is destroyed via apoptosis. CD4 cells can develop into one of three types of further differentiated cells. Recall that CD4 cells are there for you. They're there for the immune cells, the cheerleaders. And they interact with antigen-presenting cells that bear MHC2 receptor. A quick way to remember which MHC complex interacts with which type of T cell is that the product is always 8. 8 times 1 is 8. 4 times 2 is 8. So the first type of CD4 cell, if subjected to interleukin 12, is the Th1. And Th stands for T helper. So it's a helper T cell type 1. What does this cell do? Well, it releases cytokines. This is the way it talks to other immune cells and says, keep going, keep attacking, keep doing it. I'm cheering you on. Specifically, it stimulates B cells. Now, do you know what antibody B cells initially synthesize? Well, first of all, they convert to a plasma cell, but the first antibody that they make, we said it earlier, it's the pentamer, the big one, IgM. So, let me ask you a question. How does IgG antibody ever get made? Well, it's by cytokines from the T helper cell type 1. That induces a class switching of antibodies to IgG, if it's the correct and specific type of cytokine. So, these Th1 cells also stimulate and cheer on macrophages to cause them to merge their phagosome with their lysosome and start chopping up pathogens. And pretty much they stimulate every type of immune cell, including CD8 cells and other types of T cells. If this CD4 progenitor cell is subjected to interleukin 4, then it will become a T helper type 2 cell. Now, when you say Th2, I want you to think Th2 achoo, because they mediate and encourage the allergic response. How do they do that? Same way the Th1 did it, via cytokines. All right, here's a B cell here that is synthesizing now IgE. So the cytokines induce the class switching from the Th2 cell to the B cell to make IgE instead of IgM. These IgEs are then deposited and resting on the surface of mast cells, which contain an abundant amount of pro-allergenic contents. So once an allergen is exposed to the IgEs on the surface of the mast cell, there's a cross-linking that occurs and a reaction that causes the mast cell to release and expel its contents and propagate the allergic response. The final type of CD4 cell is the T regulatory cell, after being subjected to TGF-beta. T regulatory cells control all types of T cells and immune cells in general. They're kind of like the orchestrators. Time for another flash quiz. Although both arise from the bone marrow, where do B and T cells actually mature? It's a nice little mnemonic. Bone marrow for B cells and thymus for T cells.